So in the end of the last lecture, can you please quiet down, please? I think the quiz is pretty simple, isn't it? If you have a hard time with this quiz, start study. Okay, so at the end of the last lecture, we talked about the ubiquitination, and we were a little bit rushed. So let me just try to go through this process again. So ubiquitination is a process that determines what proteins are marked for degradation. And there's three enzymes that we talk about that are responsible for putting the ubiquitin covalently onto the protein target. And there's three enzymes. The first is E1, the ubiquitin activating enzyme. And you can see that the only function of E1 is to bind to ubiquitin and covalently bind to ubiquitin. And because ubiquitin, as we said, is very conserved throughout evolution, it's conserved from lower eukaryote all the way up to the higher eukaryote. Because E1's only function is to recognize a very conserved ubiquitin. Therefore, E1 itself is also very conserved. There's only one E1 in the eukaryote. Now, E2, we have about 30 plus E2s. It's a little bit more than E1 because E2 not only have to recognize E1 and ubiquitin and take the ubiquitin over from E1 to itself, it also needs to interact with E3s, and therefore their E3s are more diverse, and therefore E2s also needs to be a little bit more diverse. But then each E2 can work with many E3s, and therefore you don't have that many E2s. The E3 enzyme, which is the ubiquitin ligase, this is the enzyme that determines which substrate is ubiquitinated. So this one determines substrate specificity. And there are many, many E3s. Many times a substrate have a specific E3. So you get hundreds of thousands of E3 enzymes. Okay, so E3 is the one that contact the target. And many times the target proteins have a little sequence that's recognized by a specific E3. Sometimes it's a phosphorylation sequence, phosphorylation events that induce the binding. Others, it's the exposure of a small peptide. So E3s specifically bind to the substrate and then bring the E2s together into the complex. And that allows the transfer of the ubiquitin to the lysine residues on the substrate. And then E2, E3 dissociate from this ubiquitinated substrate. And then you start this whole process again. E1 ubiquitin comes in, transfer to E2, and E2 and E3 work together with this guy again to add ubiquitin this time to the first ubiquitin residue to elongate the ubiquitin chain. So this process, this event, so E1, E2, E3 repeat many times, and therefore you have a polyubiquitination chain. So this is how this whole process works. So this is a covalent modification, and when it's marked, this long ubiquitination chain is then recognized by the proteasome and then take the protein in for degradation. What you end up having are very small peptides or individual amino acid that really the protein is destroyed. So that's how protein degradation happens. And this process is extremely important for cell cycle regulation, which is what we're going to discuss in the next 4 lectures. So today is essentially an overview. So we talk about cell growth in size, that is the increase in protein synthesis, and then the size of the cell increases. When the size reached a certain level, it indicates that it's ready for the cells to divide into 2. So we're going to discuss the events that controls the cell from the beginning to the end. So cell cycle is a machinery, it's a process that describes the ordered series of macromolecular events, that is the protein-protein interactions that allows the cells to grow from one cell, increase in size, duplicate the DNA, and then split into 2. So this whole process, the sequence, is called cell cycle. Now the cell cycle has 2 major critical events. One is the duplication or replication of the chromosomes. So that's called the S phase, meaning DNA synthesis. So S stands for synthesis. This is a major event that need to be done very accurately with high fidelity. The second major event of cell cycle is the chromosome segregation into 2 daughter cells and the final physical division of this one big cells into two individual cells. And this is the phase we call mitosis, stand by M. So the S phase and M phase are the two major events in the cell cycle. And in between of these two are two phases. One's called, these are gap phases. That's why it's called G. G1 phase and G2 phase. G1 is the place where the cell responds to extracellular stimuli and decide whether to go into cell cycle or to stay in the resting state. And this is also the process where cell accumulate materials that's required for proceeding through the cell cycle. So this is the place where cell grow in size. So the stuff that we talked about in the last lecture, in the previous lecture, the cell grows in size, that's happening in G1. And when the cell has stocked up all the important stuff, then they can enter into S phase. This is the phase where the cell will respond to extracellular signals. All the other phases of the cell cycle are automatic. Once you pass through a certain point, it gets into autopilot. But this is a place where cell actually listen to the outside signals. Within G1, there's a little window called G0. This is a place where cells stay quiescent. They're alive, they're metabolically active, they just do not do anything. So most of our cells in the adult body actually stay in the G0. Okay? And only when you have a signal to tell cells to grow, they enter cell cycle, they get out of G0, enter G1, and start to grow in size to stock up the materials that's needed. And so this is the place where they respond to extracellular signal. Once it goes into S phase, they have to complete the cell cycle. Sometimes some cells takes a longer time, some cells takes shorter time. Mitosis usually in mammalian cells, in our human cells, takes 1 hour. S phase takes slightly longer. So majority of the time the cell spent is in the G1 and G2 phase. We don't know quite a lot about G2 phase, but we know that in this whole process, the cell cycle progression has to be, number one, very accurate so that your daughter cells inherit all the right amount of genetic material from the parental cells. The number two thing is that this event has to be happening in the right order. You cannot divide the cells without duplicating the DNA. Then what happens is that your daughter cells do not have the genetic material. Or after the cell division, you cannot have DNA synthesis anymore. Otherwise, you get too many chromosomes. You know, we all know the consequence of too many chromosomes. For example, Down syndrome. That is the chromosome number 21 has 3 copies instead of the normal 2 copies. So every time when you have abnormal number of chromosomes or abnormal cycle of DNA replication, we got genetic disease and we got the cells cannot survive very well. Or when the cells survive, the organism has troubles. So it has to be highly accurate and it has to be in the right order. So this whole process is controlled very, very strictly. And during these different phases, we also have mechanisms to check, to make sure everything's all right before you can proceed to the next step. And those are called checkpoints. And so the S phase has checkpoints, G1 has checkpoints, mitosis has checkpoints. So there are many checkpoints in the cells to make sure that your DNA is not damaged. We receive extracellular damage signals like ultraviolet light that can damage DNA. So when that DNA is damaged, the cell cycle should not proceed. So we have checkpoints to control all of these. So, so in this lecture, I'm going to give you a historical overview of how the machinery, the control cell cycles are discovered. So it actually all happened in the 1980s. It was actually by the beginning of the 1980s, people start to realize that the protein complex, the control cell cycle, are very conserved from lower unicellular eukaryote all the way to human. And it was actually very surprising in the beginning. So how come a baker's yeast would have the exactly same way to control their cell cycle as the human neuron cells, which has much higher function? But in the end, when you think about it, it actually makes perfect sense. Because cell cycle is kind of like transcription, translation, and protein synthesis. These are all the basic features a cell has to have in order to survive. And therefore, these control steps were determined and optimized during very early stage of evolution. And they're very conserved. That's why they're very conserved in all the mechanisms. So it is from this point that scientists start to take advantage of other organisms. For example, yeast. For example, amphibian eggs. Each have its own certain advantages, and it's through the work in all these systems at the same time that we figure out the most basic mechanism that controls cell cycle. So I'll talk about 3 lines of major studies. The first one is the genetic studies in yeast. Yeast grow really fast, and you can do a lot of genetic manipulations with them. So it's a great tool system. There's also a lot of study using eggs and egg extract from amphibians or marine invertebrate like sea urchins. These guys are big, and they allow you to do a lot of biochemical studies. And eventually, we have to work with human cells to make sure that everything we understand is applicable to humans. So in the yeast, the early studies were done by these 2 people. One is Lee Hartwell, who studies the budding yeast. This is the same type of yeast that you use to make bread, the baker's yeast, Saccharomyces cerevisiae. At the same time, another person, Paul Nurse, who's currently the president of Rockefeller University, he was studying fishing yeast as pongby. And the studies from both studies came out similar result. So what happens is the yeast cells are very good. They're unicellular, replicate much faster than human cells, and you can do a lot of genetic studies. So what happens is the yeast cells also go through the same cell cycle from G1, S, G2 to M phase. And if you take a whole group of yeast cells, at any given time, you have a certain fraction of your cell population in G1, in S or G2 or M. So it's a mixture of cells at every phase of cell cycle, right? It's called asynchronized cells because we didn't ask them to go into cell cycle at the same time. So they're everywhere. Now, we can make a mutation in these strands, either by treating them with chemicals or treating them with other genetic modifications. So what Paul Nurse and Lee Hartwell did is that they isolated what they call the temperature-sensitive mutant. We talk about temperature-sensitive mutant in the Drosophila case, right? So in a lot of these organisms, they're all sensitive to temperatures. So they can isolate another set of temperature-sensitive mutant, which at the permissive temperature, which is usually a lower temperature, these yeast, they're just like wild type. They grow just fine without any problem. Now, if you switch them to a higher temperature, so yeast love to grow at 30 degrees, not 37 degrees. You put them into 37 degrees, they don't like it. And for this particular strain, their mutation phenotype only show up when you put them at a restricted temperature, which is a temperature that they don't like. At this temperature, there's obviously a mutation in a protein that halts cell cycle. So they select for mutations that works that way. So in this case, in what's shown here is it happened to be a mutation that stopped the cell cycle at the G1 S boundary. So it ends up what happens is that all these cells eventually get into the state, but they lack the mechanism to go forward. So they sit there, and we call them, it's resting here. So the cell cycle is arrested here. Okay, so all these, so they can isolate mutants that arrest cell cycle at different places, at G1, at S, at G2 and M, not just here, but they can be everywhere. So they isolate a lot of mutants, and together they call these mutants cell division cycle mutant or CDC mutant. That's how the name comes from. So all these mutants now, with all these different mutant yeast strands, they can now start to do genetic analysis to isolate, to identify these mutations, and what are they? So one idea is, for example, if you take this mutant strain, okay, the idea is if you take a cDNA library from a wild-type yeast that has all the functional genes, and then you introduce them back into the cells. If one cDNA happen to correct this mutant, that will allow these cells to then keep going into the cell cycle. So in a way, it rescues the mutant. So that's the idea how people are doing an experiment. So this is an experiment. So the way they're doing it is, first of all, they're making a cDNA library or a genomic library from a wild-type yeast strain that's absolutely normal. They go through cell cycle all the time. So the assumption is that all the cDNAs or genomic DNAs from this yeast strain would not have mutations. They're wild-type. So they isolate all the wild-type cDNAs cut them into pieces. All the cDNA individual ones are cloned into a vector, put into a plasmid. Now you have a whole mixture of plasmid, each one containing one wild-type cDNA. And so this is what we call the library. Okay? So you then put this library, introduce the library, transform it into a mutant E strand. Okay, so you do it at the permissive temperature. Sorry, the permissive, what they did is 25 degrees room temperature, not 30 degrees. 25 degrees room temperature, you put this cDNA library, plasmid library, into the E cells. And you don't work with one E cells, right? You start with 1 million E cells. You put in, say, 1 million plasmid mixture. So each cell will receive one cDNA, okay? And so you do it all in the mixture. Now, for most of the cells, they receive a gene that has nothing to do with the cell cycle because, out of 1 million cDNAs, some are for other functions. It's nothing to do with cell cycle. And you don't know which one, right? So let's say in most of the cases where a cell receives a gene X or gene Y that has nothing to do with cell cycle, then that particular defect is not corrected, right? So then when you switch these cells to the restricted temperature, they will grow arrest, same way as their parental cells. And then when it grows arrest there for a long time, they die. They don't live, okay? But if you get one lucky cell that received the right cDNA, for example, in this case, showed CDC28 gene. If this is a CDC28 mutant, one of the plasmid that encodes CDC28 happened to pop into the cells, correct the mutant. Now, this one particular cell would not be arrested when you switch this to the restricted temperature. Now, these cells will keep dividing, where the majority of the cell population die off. This cell keep growing, and you give it enough time, you accumulate enough of live yeast, and all of these E cells will have the CDC28 mutant. Then you can collect this group of E cells, recover this plasmid from the cells, and then you get your gene. So it is this way Paul Nurse isolate the first CDC gene, the first gene that controls cell cycle. And this CDC28 gene turns out to encode a protein kinase. We call the cyclin-dependent protein kinase. And we're going to talk about this a little bit later, but now you just have to remember CDC28 encode a kinase. Okay? So this is what happens in E studies. At the same time, a number of other people are also doing studies in the frogs using frog embryos. These eggs are really, really big, and you can get a lot of material from them. And also is that you can control the maturation of these eggs in vitro, and you can synchronize these cells very easily. So it's very easy to get a lot of materials to identify proteins to do biochemical studies. So this is how the frog oocyte matures. So an oocyte is this cell that's produced by the frog ovary. Okay, so it's developed in the frog ovary. They replicate DNA, and then they rest in G2 for 8 months. So this is one oocyte in the ovary, in the female frogs, arrested in G2 stage and stay there for 8 months. And during this 8 months, it's not sitting there doing nothing. It actually keep doing protein synthesis and start accumulate a huge number of proteins. It's called the stockpiling. The size keep growing during this 8 months, and eventually become a really big egg, and it has all the proteins that's required for doing all these jobs. Okay, so this takes 8 months. So the egg grows really big. Now at this point, if the female egg is stimulated by a male, then the frog ovarian cells will produce a progesterone, the hormone. And that progesterone will act on this G2 oocyte and stimulate the cells to go into the first meiosis. And then it will continue to go to the second meiosis, the metaphase of second meiosis, and will stay here. Okay, so now at this point, if the sperm comes in, fertilize this egg, this is a haploid cell now, the pronucleus of this particular egg will then fuse with the male pronuclease to form a diploid cells. And that diploid cells will start to undergo 12 cycles of cell division very fast. Within a very short period of time, goes through 12 cycles using all the materials that's accumulated here. and then that divided into, so this 12 cycles of cell cycle division is very synchronized. It is 1 going to 2, 2 going to 3. And you can actually, as long as you start fertilization at the same time, all the eggs start dividing at the similar time intervals. So you can accumulate a large number of eggs at a similar stage of cell cycle. And after that, of course, then you have all these to form. After these divisions, they start to form blastula stage. And then each part of the blastula, those embryos will become different type of cells, and then they're no longer synchronized. So this process is a focus of a lot of scientists. So if you look at this stage, looks like progesterone is the one that stimulates these cells to undergo the first stages of meiosis. So the idea is the progesterone must be activating or inducing some proteins in these cells to allow them to go to here. In another word, these cells must have something that's activated that this cell does not have. So then instead of having progesterone, which has to go through all the progesterone receptors and signal transduction cascade, they decided to, let's take the cytoplasmic proteins from the cell and then inject into the cell and ask, is there a material that allows these cells to mature into these oocytes? So this is the experiment they did. And for that experiment, they take the egg arrested in the metaphase of meiosis II, take the cytoplasmic proteins, and inject it into this oocyte arrested in G2 cells. And lo and behold, at this point, even if you don't give these eggs progesterone, they start to mature, go into the meiosis part and the rest there. Of course, to go further, you need fertilization. But then they can proceed to the meiosis II stage. So this is a process that we call the maturation of the eggs. So the activity that stimulates them to go through that process is called the maturation promoting factors, or MPF. So this then becomes, this experiment said two things. Number one, that there is an MPF activity that's present, that's induced in these cells, allow this to go through the process. And number two, this is a very good assay to actually following the activity of MPF, because we know that these G2 arrested oocyte has very low MPF activities, and the eggs arrested in the meiosis II has very high level of activities. Then what about all the procedures in between? So now using this assay, scientists can now take cells from every stage of this process, not just meiosis I, but early stage of meiosis I, mid stage of meiosis I, late stage of meiosis I, interphase, early stage of meiosis II. They take cells every few minutes from this whole process, and then take the cytoplasmic extract, inject back into the cells, and ask, when is the MPF activity present, and when is the highest? And what they got is this interesting curve. So this is the MPF activity, maturation promoting factor activity. This is the time as the cell goes through these division cycles. What they found is, of course, before you add progesterone, this is the oocyte arrested G2 that has a baseline level of MPF activity. Now, when the cell goes into meiosis, this activity goes up. But then, right at the interface between meiosis I and meiosis II, the activity dropped. And then it go back up. Now, at fertilization, the cells go into the first interface before the real mitosis. The level drops again. Right before the cells start the first mitosis, the level goes back up. When mitosis happened here, the level dropped and then go back up. So it correlates with each round of mitosis and cell division. So that's why they named this activity. So they know the MPF activity must be cycling. Okay, it must be cycling as with the cell cycle. And so the idea is, how do you then isolate this activity? Because they have this assay, they think now, okay, I can take these extracts and start to purify through columns, and by following the fractions of columns, be able to purify the protein. The idea is correct, but it takes them several years. And it turns out to be actually much more difficult than they really did. And the reason is because MPF activity is not one protein. That's why they can't follow it. So at the same time, when this study was being conducted, Tim Hunt was studying sea urchin eggs. Again, sea urchin eggs are also very big, and so you can easily do biochemical studies. And they found the sea urchin egg also have this maturation promoting factor activity and also fluctuate with cell cycle. So he decided to do an experiment. So he decided to label the whole egg with radioactivity, radioactive labeled amino acid. So every protein that's being synthesized in the cells will be labeled. Okay, so he put them into the radioactivity, and then he take out cells every 6 minutes during the whole process and just run the whole protein lysate on the gel. So the idea is he asked, if there's an MPF that fluctuates, then if this MPF is encoded by a protein, then we would expect that the proteins being synthesized or the level must be high at certain stages and then goes down at interfaces and go back up in the next stage of cell cycle. It must correlate. So this is the protein gel he got. If you look at the protein B and protein C, we have no idea what they are. These proteins are synthesized evenly throughout the whole process. But then you have a protein that seems to go up a certain stage and then drop back down and then go back up again and drop back down. It seems to be cycling as the cell cycle goes. So he decided to focus on this protein, and he have enough of this protein. He cut out the band and did the sequencing and named that protein cycling, because the protein synthesis cycles with every stage of cell cycle. So this is how the first cycling was discovered. He named it cyclin B. It was discovered by Tim Hunt. And cyclin B turns out to be a component of maturation promoting factor. And it turns out the cyclin B is controlled not only at the protein synthesis level, but also the degradation. So cyclin B level, if you look at the cell cycle, this is the S phase, G2, M phase, G1, S phase. At every stage of cell cycle, as cell approaching mitosis, cyclin B level start to accumulate in the cells. Right before the cell enter mitosis, it dropped precipitously. And that's through ubiquitin targeted proteasome degradation. So this is how useful degradation can happen, because you can control the protein level really fast, and that allows the cell cycle to enter into mitosis. After the cell finished mitosis, getting to G1 at the G1 cyclin B level is still very low. The S phase, again, it starts to accumulate. So it cycles with every stage of cell cycle. That's why it's called cyclin B. It turns out MPF has two components. One is cycling. The other is that kinase that we talk about. It's cyclin-dependent protein kinase, meaning that without the cycling component, this kinase is inactive. You need the cycling to activate this protein kinase. And that's why it's very difficult to just purely purify that MPF activity, because you can't purify the two proteins by the same column. They have different properties. So once the columns separate them, you lose that activity, but the protein is still there. So that's why it's difficult. And it's through this separate effort, when everything gets put together, we understand that MPF now is a cycling CDK complex, which is a basic building block of cell cycle machinery. So it was because of this work, these three scientists were awarded the Nobel Prize in 2001 in physiology and medicine. Lee Hartwell was the one who identified the first restriction point. We're going to talk about the start or R point. And also he introduced the concept of checkpoint, that when cell cycle has problems, there are checkpoint genes that get turned on and arrest the cell cycle at that stage until all the problem get fixed. So that's a very important component of cell cycle. Paul Nurse used the yeast to identify the first CDK cyclin-dependent kinase. We call it CDK from now on. CDC28 is one type of CDKs. There are several CDKs. And then Tim Hunt is the one who identified the cycling. And with these two, that's a basic building block of cell cycle. And with a checkpoint that ensures the cell cycle proceed accurately. So these are the earlier studies that were done in the in the invertebrate and early eukaryote. And later on, from then on, then people start to use mammalian tissue culture cells to study it. And so mammalian tissue culture cells, the great advantage is you can get as many as possible. You don't need to squeeze a frog, and you can just give them serum, and they grow in a petri dish for a long time. You can synchronize these cells, and it's possible to do biochemical studies. You can do molecular genetic studies. You can introduce a lot of mutations into these cells you can manipulate the cell cycle. So a lot of studies so a lot of the material knowledge that we have now was later on based on a lot of these mammalian cell studies. So this is the cycling CDK complex. As I said, it has two components. One is the protein kinase component, and one is the regulatory. You can consider this as a regulatory subunit because it's really required for the activation of CDK kinase. And upon activation, CDK kinase can then phosphorylate substrate to initiate cell cycle events. So all the cell cycle has the component has these two components. And their mammalian cell cycles have multiple CDK cycling complexes. Depending on which stage of cell cycle the cell is at, you have different CDK cycling complexes. So we have, in the early G1 phase, we have D type of cycling D. And the CDK partners, there are two CDK partners for cycling D. One is CDK4, one is CDK6. And in many cases, they're indistinguishable. So we usually call them D cycling CDK4/6. We treat them as a similar molecule because in most cases they behave the same. They can perform the same function. So you notice that in this stage of cell cycle, the early stage of cell cycle, there's only one cycling. That's D type of cycling. Okay, so cycling D is important there. This is the part you got to remember. There's nothing you can do, but you got to know that early stage of G1 is D type cycling. At late stage of G1, passing this critical R point, then you start to have another cycling, which is the E type cycling. The E type of cycling, cycling E, partners with CDK2. It's another CDK kinase. This functions at the late G1 stage and the very early S phase. And then most of the S phase, then it gets replaced by cycling A, again with the same CDK partner, CDK2. And then S cell past this S phase going towards the G2 and the mitotic phase, you have then the cycling B. So cycling A covers this whole process, but then it has different CDK partners. Early stage is CDK2. Later stage is another CDK partner called CDC2. CDC2 name comes from the early E studies, cell division cycle 2. Another name of CDC2 is CDK1, so cell cycle dependent protein kinase 1. So people have all these genetic names, and then they try to make an effort to use one line of names for everyone, so they introduce the CDK. But then some people insist on using the old name. So you got to, sorry, there's not much you can do. So CDC2 is CDK1, okay? So there's only CDK1, 2, and 4, 6. So 4 of them, okay? All you need to worry about is 4 of them. CDK1, CDK2, 4, and 6. 4 and 6 are the same. So it's essentially 3 CDKs, all right? Now, CDK1, then at the mitosis, at the beginning mitosis, coupled with cyclin B, which is a cyclin we have been talking about. And then after that, then get replaced by cyclin D. Now, all of these different cyclins at the different stages of cell cycle, as we mentioned before, at the end of that period where the cycling works, there's always a ubiquitin-mediated degradation process. So cyclin D is rapidly degraded at this point. Cyclin E is degraded right here, and cyclin A is degraded right here, and cyclin B is degraded right here. So it's the same mechanism that controls the disappearance of the previous cyclins. And it's these control steps that's very, very important, because if the previous cycling do not go away, the cell cycle cannot go to the next stage. So you need to get rid of the interference from the previous cyclins. So all of them are degraded by ubiquitin-mediated proteasome. And then they get newly synthesized when you need it. So it's the cycle of newly synthesis, degradation, newly synthesis that controls the cell cycle. The CDK activities can be regulated at protein synthesis level as well, but mostly the activity is controlled by the presence of cyclin. Without cyclin, you can have these proteins in the cells, but they're not active. So it's mostly the cyclin expression level in the cells that determines the cycling state of the cells. But of course, this cycling CDK complex can be further regulated by other mechanisms. And let's talk about this in the next lecture, not today.